Hello and welcome to today's PIR live event webinar brought to you by Partners in Research Canada. This special webinar is for Science Odyssey. We're celebrating science all week long, and my name is Stacey Joyce. I'll be your host today. If you're joining us live, please make sure that you click the chat button anytime throughout the webinar, and you can send in your questions. That way we'll queue them up and ask them when it's time. And make sure you tell us, who the question is coming from. It could be a student's name in your class, or perhaps just the grade level and what city you're in. Whatever you want to include, we would be happy to acknowledge that it is your question we are asking when we get there. So today's guest is Dr. Teresa Stokes. She is an assistant professor in forensic science within the Department of Chemistry at Trent University in Peterborough, Ontario. We're so glad we could have you with us today, Teresa. I will let you take it away.
All right, thanks, and hi everyone. I'm just gonna load up my PowerPoint here. And I'm really, really excited to be here and tell you guys a little bit about a field that I'm very passionate about in Canada. So we're gonna talk a little bit today about forensic science, what it means to be a forensic scientist, and you'll take a little screenshot in my world in forensic research science. So we'll look at what's it like to be a forensic research scientist in Canada, what it's like to be me, and what kind of my days are like looking at blood and blood stain pattern analysis. And then you'll maybe see some special guest appearances from some of my students in the lab. So they've graciously donated some of their time today to tell you what research projects they're working on here. So thanks again, Partners in Research Canada. I'm really excited to be here. I'm Teresa Stokes. I'm at Trent University. And if you don't know where Trent is, it's geographically between Toronto and Ottawa. We have a great forensic program here and chemistry program here. So if you're interested, come on in and check it out. Okay, so today, just quite quickly and briefly, I'll introduce the concept of forensic science to you guys. More specifically, we'll look at blood stain pattern analysis, how it fits within forensic science, and also how there's a push towards making synthetic blood or fake blood. If you've seen in the little webinar demonstration or kind of outline of my talk, what's it like to make fake blood, and why in the world do we even care about making fake blood for crime scene investigators? I'll introduce briefly some of one of the exercises that is also posted online. We do this a lot in different secondary and high schools around the Peterborough and Kawarthas area. So these are really interesting and fun experiments, and they can show you how much science, chemistry, physics, and biology is linked in blood stain pattern analysis. Hopefully I'll have video to show you some demonstrations from my students. And of course, I will be happy to take any questions that you guys have about forensics and my research. Okay, so forensic science. Most of you guys, if you're interested in forensic science, you may have seen CSI, maybe you've watched Dexter, NCIS, or something like that. And I'm here to kind of unfortunately blow away the myth that forensic science is very glamorous. Unfortunately, we don't walk around crime scenes in our heels. We don't wear sunglasses. And for the most part, we actually look a lot like this. In our bunny suits, we've got face masks on, we're sweating up a storm, but we're doing good work. In terms of collecting traces and understanding and identifying traces and how it works within a crime scene and within a legal context. So first and foremost, when we talk about forensic science, we're talking about it as being an interdisciplinary science. And you'll see, like I mentioned before, how many different core and fundamental sciences are actually integrated into the idea of crime scene investigation and evidence processing and analysis. So specifically, it involves the study of traces, and in this case, to support legal investigations. And here are some major players in the field of forensic science. So all the time, if you're in a career in forensic science, you may be communicating with the police, laboratory specialists, lawyers in the justice system, and academics like myself. So it's very important then to put that there's clear and concise communication between all of the different stakeholders in the field. So you'll see we do sometimes use jargon terms, but for the most part, we have to communicate with a variety of different sources. So I'm here in the research world down in academia, and in my day-to-day research activities, I communicate with both the police, and mostly laboratory staff. I personally don't communicate much with lawyers in the justice system. However, there are lawyers here on staff at Trent where we're looking into some interesting research projects there too. Oops, okay. So what exactly is a forensic research scientist? So like I mentioned, we basically do applied and practical research in forensic science. So here in Canada, there's a small group of us that do core specific forensic science research, but it's really fascinating stuff. So there's lots of work in forensic anthropology, entomology, so the study of bugs, fingerprinting, toxicology, genetics, and then here at Trent, we have a group that works on blood stain pattern analysis, and I'm one of those members of that group, which is really exciting. But overall, in terms of kind of mission statements for research here in Canada, what we do is most of the time support frontline members. And when I talk about frontline members, I'm talking about organizing or people, police officers or people in the lab or lawyers. So all kind of in that little diagram I showed you before. We're actively involved in any sort of consulting and training. So sometimes I get to have the really nice opportunity of being involved with police training, which is quite exciting. And we do, sometimes we get asked to be invited in groups that are heavily dominated in policy and decision making. And so for any of you guys who have actually looked into the state of forensic science in Canada and throughout the world in the past few years, you'll notice we've taken a little bit of a hit in terms of criticism on really integrating fundamental forensic principles in our science. So we as research scientists try to address that all the time. So one thing I always get asked when I talk to people about my research, the first thing people say is, "Oh, you're like Dexter, like that show Dexter." And unfortunately, as much as it's really cool, in terms of the media, I'm more like this Dexter. For any of you guys that have actually watched Dexter's Lab, that's who I identify more with. Okay, so blood stain pattern analysis, it's a sub-discipline of forensic science. Now, in terms of definitions, if you're interested in definitions, blood stain pattern analysis, it's the study of the size, the shape, and the distribution of blood stains that could be found anywhere, particularly at a crime scene. And they're used to give kind of determine the bloodletting events. So what are the physical and mechanistic events that gave rise to the origin of the blood stain pattern? So behind this slide here, you'll see that behind us we have what's one of the types of patterns. It's called an impact pattern, noticing that you have lots of blood stains of all different shapes and sizes. Now we as scientists use methods where we can actually pinpoint some specific blood stains. You'll see my mouse kind of floating around, hopefully. And we use the shapes, and I won't get into it in so much detail, but we can use the shape and their actual positioning to figure out where the blood source was and if it came from one single event. So we do use a lot of math, physics, and fluid dynamics when we look and try to analyze some of the patterns that are found at crime scenes. This kind of work eventually could make its way into court and kind of, again, determining what could have potentially happened at a crime scene. So for those of you that are interested in blood stain pattern analysis, or maybe you're thinking about going to university in forensics shortly or in a few years, if you're looking at embarking on a career in blood stain pattern analysis, it's important to know that you should probably take a lot of different sciences because we rely heavily on fundamental principles from a variety of disciplines. For example, when I just talked about the impact pattern, we rely on the physics, the fluid dynamics, and physical principles of fluids and how they behave in different scenarios, and particularly how in different environments of force. Mathematics to actually go in and figure out how these blood stains travel in time and space. We use biology. One major deliverable from blood stain pattern analysis not only is the kind of identification and interpretation of these blood stains, But also, the police officers will quite often take swabs of blood that are found at crime scenes, send it to the lab, and from there we'll obtain a genetic profile to identify an individual or a bleeder at a scene. So we do tap into the worlds of biology. And in terms of chemistry, you'll see today we're going to look at what are some chemicals that we can put on a crime scene to see maybe if there's blood there or not. So we care a lot about the chemistry there. And in my world, in my background, engineering and material science, because it's quite interesting new addition kind of to that field, because we need to understand the material and engineering properties of blood and blood stain events in order to get a good understanding of the fundamental science behind the field. So I won't read this slide to you guys, but basically all I wanted to let you know is that where my research kind of falls within the scope of forensic research and blood stain pattern analysis is really an integration from a materials perspective on blood stains. So I argue, and a lot of people, we all agree, that blood's often left as physical evidence at a scene, and it has measurable and reproducible characteristics that could be very useful to crime scene investigators in terms of their reconstructions, any sort of research or training that they are involved with. Now, if we look at blood from a material perspective and just hone in on what are specific properties of blood that are quite useful for blood stain pattern analysts and investigators to use, perhaps we can also make blood or fluids or blood analogs that mimic blood and do the same thing. So there is a need in the forensic community to actually make blood substitutes, because blood's sometimes really difficult to work with. And I'll show you in a slide in just a bit some examples of why using blood is kind of icky and difficult to work with. So we in the lab look at making a lot of fake blood products that can make the lives of forensic investigators and blood stain pattern analysts a lot more easy. So I just wanted to show you a video that my graduate students put together on what we actually do. So this is just a little clip. It was submitted for a video contest.
Blood. In forensic science, this unique fluid can tell us a lot about a crime. The more we know about blood's behavior, the more we can learn from a scene. We use blood to study blood. Now what if I told you that this isn't blood?
At Trent University, members of our research group have developed a forensic synthetic blood substitute, the FBS. This fluid not only looks like blood, but is designed to act like blood, providing a novel material for forensic research and training.
Previous work demonstrates that the FBS mimics the complex properties of blood. Students, researchers, and police agencies are using this material to create and study blood stain patterns.
In forensic investigations, chemicals are used to uncover blood stains. We've designed the FBS to do the same. We are also working on adding genetic material into the FBS for use in forensic DNA analyses.
Our fluid is a safe and ethical alternative to using human or animal blood. With this material, we can train current and future forensic professionals.
I'm Amanda Orr.
I'm Samiko Polacco, and we're making an impact in forensic science research.
All right, so that's just a little video from my research lab and just a little snippet of what we do. So just in summary, what's the need and what's the driver for making blood substitutes? Well, like we mentioned, blood's sometimes difficult to work with. It's considered a biohazard, so sometimes when you want to use chemicals that maybe aren't involved with any sort of health and safety risks, for example, the transmission of blood-borne pathogens, blood, it's in us. We know that if we cut ourselves, if you've ever, accidentally tripped and fallen or maybe experienced a little cut right away, your body's built mechanistically to start clotting and start preventing itself from all the blood escaping your body. So we in forensic research, in the research world, find that quite difficult to work with, because we only have a few seconds to a few minutes to actually work with it, to have the properties of the blood that we actually care about in a crime scene context. So if you can make a material or alternative that actually has a long shelf life, meaning that you can put it on the shelf, have it stored for a couple months, and the properties will remain the same, you're doing a really good job in terms of making blood substitutes that are effective for crime scene research and investigation. So things that we make fluids that don't really have clotting factors. The blood is known to hemolyze, so the cells can burst in different conditions, which will again change the properties and make research a little bit more difficult. And having materials that don't have, say, red blood cells or anything in them can reduce the chance again of acidosis, or again, a phenomenon that can alter the properties of blood. making blood-free or synthetic blood substitutes are quite cost-effective. They bypass the needs for ethical approval. So sometimes, for example, in institutional organizations or even universities, it takes a long time to sometimes obtain ethical approval to actually use blood. So that's a kind of an advantage for that. And for me, kind of the most important part of my project is to ensure standardization of scientific principles and properties within the field. So if you have a fluid that has known and reproducible properties, then perhaps you're doing good science. That, while paired with good scientific methodology, can give you really nice research projects in the field. Okay, so what makes a good synthetic blood substitute, Teresa? What are you talking to me about? Well, it depends on your outlet. So, for example, the medical professionals want to make blood substitutes that are really good and effective at transporting oxygen throughout the system. We move to the entertainment industry, we're talking about blood just looking good, right? But in forensics, we want to make sure that the blood behaves like blood in a physical sense, a chemical sense, and a biological sense. So today I'll talk to you quickly about the physics and the chemistry behind blood substitute development. So we've done a lot of research with our group, and here are four, when you go and you start thinking about making your fake blood and for your experiment, here are some things that you should think about. Your material should be safe, preferably free of biohazards. We spend a lot of our time in the research lab suggesting that blood substitutes should hold scientific validity in a relevant forensic context, meaning that the properties that you care about should be standardized and measured. From a material science perspective, we say the blood substitute should be easy to make and easy to use, either in the classroom or at the laboratory. And for us, we spend a lot of time doing neat things like offering new and improved ways to think and learn about crime scene investigation and blood stain pattern analysis. So for example, we can make some blood substitutes that glow in the dark. So you can put a bunch of patterns on top of each other, ask crime scene investigators to perform an analysis, and then we can shut the lights off and say, okay, did your analysis actually meet the expectations? Where are the limitations of the field? What can you actually learn from these procedures? So it's quite an exciting time to be in this field. Let's look at point 2, the scientific validity. So here are some things that my research has specifically focused on, building fluids that really mimic the physical properties and physical characteristics of blood that are really relevant to blood stain pattern analysts in the field. So here are 3 properties that we care a lot about. We care a lot about viscosity, so the fluid's resistance to flow, the surface tension of the fluid, and the density of the fluid. So we try to make fake blood that has the same characteristics of whole human blood. They're quite important in the way the blood actually behaves in a relevant crime scene context. So here's a curve. This is typically a result that we would look at in our lab. It's called the viscosity profile of one of our materials. I should mention before, our materials are made from, we use polymer chemistry to actually make our materials. They're inorganic, organic silicon sol gel materials. So if you want to look that up for homework, maybe you can do that too. But anyway, here's an example of viscosity profile. Now, it's really important to know that blood's viscosity isn't constant. In fact, it actually changes, and it thins, it gets less the more force or the more shear that you apply to the blood. So we spend a lot of time making sure that, here's just examples of our materials under different temperature conditions, that these materials also exhibit shear thinning properties that blood has over the course of how much shear you actually put into the material itself. And so this example, this curve, I could put also here that the whole human blood range and our materials actually fall within that expected range and observed range in the literature. When we look at things like surface tension, you'll see in a video that Mitchell was talking a little bit about surface tension. Hang tight on that video, but we can actually go in and calculate the surface tension of a droplet based on its contours just before full droplets form. So you're looking essentially at fluids. Don't worry too much about the names here, but let's look at H2O or water, for example. So this is an example of a snapshot of water that's being released from a glass pipe past your pipette tip. And now just before the droplet forms, we take a photograph of it, and we can use the shape and the contours to actually calculate the surface tension of the fluid. And we make sure that our FBS falls within the human norm. I think the human norm is somewhere around 54 to 61 millinewtons per meter. Here's just a summary chart quite quickly. One thing that we do to actually validate our materials and compare them to commercial standards or other fake bloods used in crime scene investigation or the entertainment industry is to see how blood light they actually are versus, and we also do a lot of our comparisons to water, which is rheologically, when I use that word, I'm talking a little bit about those viscosity profiles, how different it is to a Newtonian fluid like water, and blood's a non-Newtonian fluid. So here's just an example of that. So these acronyms here, LAGT and FBS, or a forensic blood substitute, have similar profiles or properties to whole human blood, which is different than water. So what you'll be doing in the lab is looking, or just thought I'd introduce quickly the experiment that Stacy really graciously put together. So when you make your fake blood, you can look at things like how does the dripping height affect the size and the shape of a blood stain. So you can take an eyedropper with your fake blood, form a droplet, maybe have it fall on a piece of paper or something, and see what actually happens when you change one variable like dripping height on the size and the shape of the stain. So you'll look at things like the diameter of the stain, so you can measure the diameter of the stain. This is an example of a passive drip stain, so it was dripped from an angle of 90 degrees from the surface. So you can look at things like how the diameter of the stain changes with increased dripping height or even impact velocity if you're in a physics class and you want to calculate impact velocity. And you can also look at how these little fine features called spines and scallops also change with increasing impact velocity or not. So here's an example of a nice little spine here. These little these little stains here are called satellite spatter. And then some of these, the more scallopy looking convex features are scallops. So if you want to go even further, you can even plot some of these results and use put on your scientific data analysis cap and look at and actually plot how does the size of the drip sign diameter, does it increase or decrease with dripping height, and how so? Is it in a linear fashion, a non-linear fashion? Are there influences like terminal velocity that affect the size and the shape of the stain? One thing I'm going to quickly talk about before I play this video, because I think I'm running out of time, is how the chemistry requirements. This is a brand new research project that some of my students are looking at, and I'm really excited to share this to you, with you. So if you've ever watched CSI before, and you'll have seen some of the investigators spray chemicals at a crime scene, you'll see this blue glowing, and everyone goes, wow, there must be blood at the scene. Well, this chemical is called luminol. It essentially, it's in a basic environment with hydrogen peroxide and another base, typically like sodium hydroxide or something like that. And the iron in the hemoglobin found in the red blood cells, actually catalyzes the decomposition of luminol. And during this process, you end up having a luminol that goes to an excited state, releases light as it returns back down to a ground state. And during this process, the light that's actually emitted falls within the blue light spectrum. So our work, we spend a lot of time characterizing the luminol reaction and making fake blood substitutes that will react with luminol the way that whole blood does. So what you're looking at here is just an example of our blood stains. Typically, crime scene investigators will spray this chemical at a scene for very diluted blood or latent blood, which means that you can't really see it with your eyes alone. So we've made, and here's an example of our fake blood on Q-tips, sprayed with the luminol, and we do a lot of work at really testing how bright the chemiluminescence intensity is, the half-life of the reaction, the duration of the reaction, and we spend a lot of time optimizing our materials so that they behave just like blood and not other reagents. Okay, so I'm going to end with just a summary. Yep.
I'm silently in the background here for everyone who's watching the webinar, giving a timeout symbol. I'm just mindful of the time. I did want to make sure that within our original sort of 30-minute quote for our webinar that we had a chance for questions.
Okay.
So I thought that we might take a pause for a few questions now, and then we can watch the video after that. And if anyone needs to leave the webinar and to go to another class, or because that they didn't plan to stay later, we will make that video available afterwards so that you can watch it at a later time.
I'm sorry I talk so much.
it's so much great information, but I thought this might be you know, a more efficient maybe use of our time.
Yeah.
So the first question I have is, how much math and physics do you need in order to become a forensic scientist?
Okay, well, I would definitely say you definitely need, I would take up to your grade 12 physics. We spend a lot of time when you look at the physical principles, knowing a lot of kinematics, in my world, fluid dynamics, but when you think about forensics, everything from accident reconstructions to understanding, say, bullet trajectories, you need to know a lot of physics. And that goes hand in hand with taking mathematics, calculus courses, and statistics. Statistics is a big world in terms of, it's very important for the way that we report our evidence to the courtrooms. There's a big push to trying to figure out the best type of statistics to actually use and say that this evidence, and kind of explain what the scientific evidence means to a jury. So I would say you're gonna have to suck it up, guys, and take a lot of physics and math. I think it's great. I think it's really fun. So hopefully you guys too.
Wonderful. We have a couple questions now specifically about your forensic blood substitute. And the grade 6s are wondering, what do you use to make your fake blood in the lab?
I should have mentioned that a little bit better. So in my research world, we use a concept called sol gel chemistry. It's called solution gelation chemistry. And we make inorganic polymers with a silicon base. So for you grade 6s, that just means I make my fake blood out of a liquid glass, okay? So you can't eat it, you can't put it in your body, it's definitely not good for you, but this little liquid glass polymer does a really good job at mimicking those physical properties, some of them that I talked about today, and chemical properties of blood. So it's a really versatile material. We can put in a lot of chemicals to make it behave more like blood, react more like blood, look more like blood, but our base material is essentially a liquid glass.
Wonderful. And we now have a question from Calgary wondering, in Mr. Mack's class, what makes the synthetic blood red?
Okay, excellent. So one of our iterations of the synthetic blood, it has iron citrate in it. So we do have an iron base. The chemical that you saw that reacts with the luminol has horseradish peroxidase added to it. It makes nice diluted kind of blood looking stains. And then sometimes you guys, you can just get away with putting a little bit of dye in there too. So that just making sure that you do the science, you do take your measurements and say, hey, the dye or the paint or whatever you're adding doesn't necessarily change or influence those properties in a way that makes not realistic blood stain patterns.
Right. And so how long does that take for your group to make FBS?
All right. So it takes about a week from start to finish. The building of the polymer system requires a bunch of stages in terms of adding chemicals, removing some solvents, and then resuspending them in other solvents. So it takes us about a week, if we're really good, maybe less than that, to make a batch of it. And we can make anything from a few milliliters up to a couple liters.
Lovely. And Isaac in Calgary is wondering how the blood stays liquid if it is made from liquid glass.
Ah, very good, Isaac. Good question. So we've spent a lot of time really making sure that that liquid glass stays in the liquid form for a really long time. So the shelf life of our materials is a few months, but you're right, Isaac, in the fact that eventually they are polymer systems. So they will harden and they will make solids over time. And that's kind of a beautiful benefit to our fake blood, is because the stains, once our fake blood has hit the air and hit a paper surface or other kind of surface, right away the stains will dry. they'll dry in a fashion similar to blood. And then eventually, if you wait even long enough, they could oxidize, change colors, and start cracking and kind of flaking the way blood would, or aged blood would at a crime scene.
Really interesting how similar you've been able to make it in all of those ways that blood behaves from a forensic perspective. Obviously not the same biological functions. It's probably not carrying oxygen throughout its vessel, but let's go with a more broad question now about your role. What made you choose to be a forensic research scientist?
Okay, so if you hear a lot of podcasts in forensic science, you'll find that a lot of people kind of stumbled into the field. You either have people that are like, I love CSI and I want to be a forensic scientist, or you'll be like me who kind of just stumbled into it. So when I was in your guys' shoes, trying to figure out in university what was I going to do, I couldn't make a decision. So I said, hey, forensics. Hey, I like chemistry, I like biology, I like math, I like physics. Look, I saw a forensic program here at Trent. Actually, I did my undergrad here. And I said, let's take it. So how I got involved in bloodstains was I took a crime scene course here. And at the time, who's still a professor here, Mike Illis, he was a staff sergeant of the Ontario Provincial Police, sent me a message and saying, can you come to my class? I said, or my office, I said, okay, sure, yeah, no problem. And he said, would you like, to work as a research scientist with the OPP for a couple summers? I did, and I fell in love with the field. I think for me, it's a really interesting field. And so, yeah, that's how I kind of fell into forensics. And I've been lucky enough to kind of go around the world and help with training programs around the world, do some research and scholarship around the world in forensics. So it's been a really interesting, kind of fruitful career for me.
Wonderful. And do you collaborate with anyone typically in your research?
Yep, we do. We have a lot of collaborations with police organizations in Canada. We also have a lot of academic collaborations within Canada and externally throughout the world. So for example, some of the blood substitute last year made its way into a training course in Algeria. So that was kind of an interesting project that I worked on. But yeah, we have collaborators in New Zealand. I'm trying to think off the top of my head. It's made its place in Europe, North America. The forensic community is quite small. The research communities were mighty, but we're even smaller. So we really value being able to communicate and collaborate with each other. So if you're thinking about a career in forensics or forensic research, you'll find that it will take you around the world and you'll have really interesting experiences.
Well, that does make it sound pretty appealing. I'm not going to lie. So do you have any advice for students who might be interested in this field?
Right. Yes, I do. Stick with it. Don't be surprised if it's not like the TV show. Keep sciences in your future if you really want to stick with a forensic science and a lab background. See if you can take a tour of a forensic facility, or maybe if you can, talk to a local forensic identification officer. It's always good to get those experiences in early to see if that's exactly what you like to do. And for me, I think the biggest piece of advice that has kind of helped me carry through my career is just to be creative, be open-minded. So we're a relatively new discipline, so that means our playground is quite unexplored. So if you have a very creative cap and you like science, I think you'll find that there's a lot of projects that can be done, and there's a lot of projects that probably should be done in forensics.
Wonderful. Well, I'm going to turn things on their head a little. We're going to do our sign off now, and then we'll play the video at the end for anyone who can stay. So before we play that video and maybe lose sight of it, I want to make sure that we thank you so much, Teresa, for joining us today and giving us an insight into the world of real blood stains and fake blood stains and studying all of it along the way.
Well, thank you guys. I really appreciate it too. This is very cool and a very good experience.
So more information about any future PIR live events or any other PIR educational programming is available at pirweb.org. And today we're going to leave you with a video of the blood room at Trent University.
Okay. All right, everyone. Well, welcome to the blood stain room here at Trent. This is just one of our research labs. I thought most of my students, my mighty research groups here, working this morning. So let's just interrupt them. Let's see what they're up to today. It's gonna be some really fun, I'm pretty sure, some really fun and exciting experiments going on, okay? So let's check it out, the blood room. Oh no, it's locked. Hey guys, how's it going this morning? Good. All right, so we're here with Partners in Research Canada, and we basically just want to know what you guys are doing, what you're researching, and you know how you're making a difference in the field of forensic science research in Canada. So let me introduce everyone. So this is Mitchell. If you want to wave, this is Mitchell.
Hi there.
Mandy and Samiko in the back there.
Hi.
All right, guys. So we'll go around in a group. Why don't you guys just tell us what you're doing, what you're researching here in our lab?
So like Teresa said, I'm Mitchell, and it's my first 2 weeks here. I'm an undergrad student at Trent University, and currently I'm looking at fluid dynamic properties of the blood substitute, measuring surface tension, and in the future, hopefully going to be looking at peroxidase-based and peroxidase-like reagents that can be added to the forensic blood substitute.
Awesome. And so is this your setup here, Mitchell?
Yes, it is.
Cool.
How it works is you take, with the camera and with this pipette, slowly drip the blood substitute into a tray and taking pictures and then analyzing it on the computer.
Oh, so right, the surface tension of the droplet, it's influenced by the shape of that droplet, right?
Yes, it is.
Oh, cool. Well, we really look forward to hearing about your results. Thanks. All right, let's move over. Let's move over Pano here and see. Oh, we might have to reduce the height a little bit. Sorry, Mandy. Don't worry, I'm the same height. So,
Yes.
Mandy, tell us what you're doing here. What are you doing at Trent?
So I've been here for almost a year working on my master's degree. So I'm working with the forensic blood substitute as well. And right now this is one of my side projects where I'm looking at how the FBS oxidizes over time, or how it changes color over time.
Okay.
So this has been something I've been working on since January, and we're looking at the color changes in our blood. So this is the FBS at almost 5 months of aging, and so when it first dropped it was very bright red, and we can see the color has changed quite a bit. So that's what I'm doing right now.
Fantastic.
I'm also working on, the main part of my project is to work on adding DNA into our FBS.
Oh, and why would you want to do that?
So we do that so we can use it for research and training analysts, anyone who's interested in the forensic DNA analysis field, on how to analyze like complex mixtures, and we do that so it's a safe way to give students an experience on how to analyze DNA samples.
Oh, very cool. So you like researching here?
I love it. It's a lot of
perfect.
fun.
Awesome. And then we'll move over. Let's move over to Samiko. Hi, Samiko. We're going to come over a little bit and see. It looks like you're looking at a blood stain pattern here, an impact pattern.
Yes.
Is that correct? Yeah.
That is. So this is an impact pattern on the wall over here, and we actually created with this very cool impact device, and it comes with a little trigger box. So we take a sheet of paper like this, put it behind there, put a little bit of our FBS or our sheep's blood, depending on what we're doing that day, into the petri dish, and we press the button, and it creates a pattern that looks like this. These patterns are pretty cool because you can actually have students analyze them and work back to the exact place where we tried to where we had that petri dish and created the pattern. So it's kind of useful in a forensic context because you might be able to tell where your blood source or your person was when this bloodletting occurred. So these patterns are something that I worked with in my undergrad when I started like Mitchell, but right now I actually work on a luminol reactive FBS. So you guys might have seen luminol before on TV or at CSI. It's this fluid that they use, and they spray it around at crime scenes, and you will start to see blue chemiluminescence or blue glowing, and that can indicate to an investigator that there might be blood there.
What's the reaction? What's going on there?
So what's actually happening is luminol, part of the solution, has hydrogen peroxide in it, and the hemoglobin that's in blood acts as a peroxidase to sort of decompose that hydrogen peroxide. And so it's what we call a chemiluminescent reaction, so there's light being emitted, but there's not heat being emitted from that reaction. So what I've been doing for the past 2 years or so for my master's project is working on creating a synthetic blood substitute, or having our blood substitute react just like blood does to luminol. And I do this by adding horseradish peroxidase into it. So that is a true peroxidase, which will decompose that hydrogen peroxide. And when we put it into our synthetic, we can actually mimic some really unique blood-like characteristics. And if you guys want to look at that, we can go and take a look at how that works.
Oh, we definitely want to see how this works in real life, real time.
Yeah. Okay. So what we have here is a piece of cardstock that has a transfer pattern created using our luminol reactive FBS, or our HRP FBS. Right now you probably can't see too much on it, so by using luminol and spraying it at a crime scene, investigators can potentially uncover bloodstains that are diluted, so someone might have tried to wash them away, or that are on dark surfaces, such as, you know, your jeans and things like that, so it's a lot harder to see. So by spraying luminol, we'll actually be able to illuminate this particular pattern on this cardstock here. So in a second, I'm going to shut the lights off and then spray luminol, and you might be able to observe some chemiluminescence. One thing to keep in mind, though, is that this pattern was created for demonstration purposes under laboratory conditions, so you will not always observe a pattern that looks like this and the amount of detail we might see at a crime scene. All right, lights. So what you're observing here is our HRP FBS reacting to the luminol that was sprayed on it. And when these two materials come in contact, energy is released, and that is released in the form of light, but not heat. So that's why it's called chemiluminescence, and that's what you're seeing here, this blue glow.
Awesome. So that's the video.
Wonderful. Well, we hope you enjoyed the live event today and the video that we appended, especially at the end. And we hope you join us again soon. Thanks, everybody. Bye now.